And this particular project focuses largely on our ability to study the evolutionary processes of early cancers. And with that, and intrinsic to that, is our ability to detect cancers at their earliest stages of development. So I just wanted to, right at the beginning, highlight my collaborators on this program. And let me know, by the way, I'm having a bad Zoom day. Sorry, both either Zoom or Wi-Fi day. So somebody, well, I guess I'll be the first to know if things start flickering or something like that. But somebody will have to ping me, so I'll keep my phone up here. So I just wanted to highlight my collaborators, as I mentioned. So Daniel D. Carvalho and John Dick were the two individuals that we initially proposed, where we initially proposed the program. And during the development of this program, we brought Scott Bratman and Segi Abelson into the program as well. The people who are actually doing all of the work, I should highlight right from the beginning, are Kimberly Skeed, Nicholas Cheng, and Alyssa Bader. And so for the projects that I'm going to be talking about, AO Project 1 and AO Project, Sub-Project 1 and Sub-Project 2, those are being led by Kim, Alyssa, and Nick in that order. Now, one of the reasons that we've decided to take this project on is largely because, well, we don't really know a whole lot about early detection and early evolution. We've heard a lot about early detection, and there's a lot of industry and research hype right now in that space, where we're spending a lot of time and effort as a community to try to detect early cancer signatures using a number of biomolecular approaches. And one of the reasons why this is such a critical activity in cancer is because our ability to treat cancers certainly improves as the earlier we can detect a cancer. And for many of the cancers that we're talking about here, they're often detected and diagnosed very late in their progression, and so that has a dramatic impact on survival. For example, lung cancers. Most lung cancers are diagnosed very late when things have metastasized and the tumor and biopsies. When the tumor has progressed fairly substantially. And of course, the impact on lung cancer of that is that lung cancer is one of the more predominant diseases contributing to deaths worldwide. So one activity that's taking a good head of steam at the OICR is our ability to use cell-free DNA or circulating tumor DNA to detect a cancer from biologics. And one program that I've been leading for the last 3 or 4 years now is a program that's looking at sporadic cancers. And Trevor Pugh, with his colleague Raymond Kim, is focusing in on hereditary cancers. And in either case, we're using similar technologies to be able to capture these biologic signatures from both cell-free DNA and circulating tumor DNA. And one of the things that we're hoping with what we can do with this is try to capture these signatures in participants and or patients at their earliest stages of development, not only so we can develop tools or biomarkers that can capture a cancer signature earlier and changing the standard of care in this space, such that we're capturing it perhaps multiple years before these cancers progress, but also so we can get a better handle on what is the biology or the genome biology in this space, because most of our understanding of genome biology of cancers is largely coming at the time, it might even be stage 1, stage 2 for, say, breast cancer, but that's still captured at the time of diagnosis when there's already there's already that progression. And so we're trying to see if we can see that signature earlier than that traditional time of diagnosis, and then say something interesting about what does the genome biology look like at that earlier stage. So we have 2 projects in the AO Project 1, Early Cancer Project, and they're broken down into 2 sub-projects. So here are calling them Project 1 and Project 2. One is looking at largely liquid, or if you like, blood malignancies. So that's Project 1, where we're looking at the role of age-related clonal hematopoiesis associated with blood malignancies. That's a project that is largely being led by John Dick and Segi Abelson. In project 2, we're largely looking at solid tissue biopsies. And when we first started this program, we were thinking about focusing project 2 on lung cancers, but actually we've expanded it to more pan-cancer focus. And that's what you're going to be hearing about today. So our early detection workflow is largely working in this liquid biopsy space, whether it's in the blood malignancy space or in the solid tissue space, where we're trying to capture these molecular or methylation epigenomic features that might allow us to detect these early signatures and then develop robust computational tools such that we can be more efficient with our multi-cancer screening approaches. Now, one of the things that I want to highlight with this project, I'm going to talk about this a lot more tomorrow, would be to focus in on the cohort. So tomorrow there's a town hall meeting for OICR. I don't think I've ever been to a town hall meeting for OICR, but I think largely we hear all kinds of news about the OICR, but I'm giving a 10-minute or an 8-minute talk then. And we're going to be talking a little bit about how population cohorts tomorrow can support early cancer research. So I'm going to talk about it today in greater detail, in fact. So this sort of gives me a sense of what's going to work and what's not going to work tomorrow. So the cohort that we're capturing biologics for these studies today is the Canadian, it's changed and the slide is a little late actually. It's the Canadian Partnership for Tomorrow project, where we collected biologics from 320,000 Canadians, now 333,000 Canadians from almost all provinces across Canada. And what's cool about this cohort is that we have biologics from people who came into the cohort healthy and may have since been diagnosed with a cancer. And so that gives us that, if you like, that snapshot of that window by which we can do that exploration or that investigation. And that's largely what I'm trying to show you here. We might have a person coming into the population cohort, say today, and 5 years from now, 10 years from now, 15 years from now, they then develop a cancer. But if they came in today, or maybe they came in 5 years ago, gave us a blood sample, that blood sample would have a signature, might have a signature in it, because that person hasn't actually been diagnosed with that cancer, and we can explore that biologic and see if we can capture that signature of that molecular cancer. So in order to do that, we've been doing these activities where we link that population cohort, say in Ontario, to a registry like the Ontario Cancer Registry, and then we see if that participant that came into the Ontario Health Study or the Canadian-wide study has then since been diagnosed with a cancer after they've come into the cohort. So we started with Ontario, of course. Kim largely led this activity here where we took samples, and this was largely done as a project through my lab. That's why you see Awadalla lab there on the top left. Took the participants from the Ontario cohort, gave the identifiers to Cancer Care Ontario, where they linked the participants, told us who was had a cancer both when we came into the cohort, so it helps validate the information we got from the participant, and also then told us who had since been diagnosed with the cancer as well. And so this is the kind of information we can capture. So Kim put this figure together for me, where what we'll have is like on the x-axis, right, on the x-axis on your right, is a figure where we say the age of the event, so the age of the participant either coming into the cohort, giving us a blood sample, filling out a questionnaire, and then since being diagnosed with that cancer. And then we have these time to diagnosis. So if somebody came into the cohort before they were diagnosed, you will see a red dot before a green dot, right? And so that's where we can capture those incident cases, and there's where we can start exploring for looking for molecular signatures of early cancer. And so right now we focused on the left, you see a Venn diagram on, this is the numbers we've got as of 2018 maybe now for incident cancers. In fact, I think this number has actually grown, and we're actually expanding this to also include many other cancers as well. So the first project I'm going to start with are the blood malignancies. So I'm going to talk a little bit about what we're doing there. I've talked about this already a fair bit, so I'm going to do a kind of a superficial pass of this because I really want to get to project 2, because that'll be the first time I've presented this to this group. But project 1, we have heard me talk a little bit about before, and so I'm just going to give a kind of bit of a refresh. So in project 1, we're looking at the impact of what we call these early signatures of cancer. But here we know what those early signatures look like, and for a lot of blood malignancies like acute myeloid leukemia, a process known as age-related clonal hematopoiesis is considered to be the initiating phase, if you like, or it's a condition by which you need to have ARCH before AML actually progresses. Now, it doesn't mean that if you have age-related clonal hematopoiesis, you will develop into a cancer. But what it does mean, though, is that you do need to have that if you are going to develop a cancer. So we now, in a population cohort, whether it's the Canadian cohort, like I talked about, or the UK Biobank, or other cohorts in Europe, we have a lot of blood samples. And so these cohorts are perfect for looking at whether somatic mutations are accumulating in blood and whether individuals progress to cancer or not. So in our first project, we're looking at age-related somatic mutations occurring in stem cells. We're looking at their impact on the transcriptome. We're looking at single-cell assays, which you're going to see about today. And we're then also going to talk a little bit about how we use that information together to understand or model the evolutionary scenarios that we think are acting on those somatic mutations. So what is age-related clonal hematopoiesis? Maybe this is a better place to start. So age-related clonal hematopoiesis is largely a function of somatic mutations that are occurring in bone marrow stem cells, which may then lead to either in the mature blood pool, either immature white blood cells relating to myeloid leukemias, or they may not, and are actually fairly non-malignant and or neutral, if you like, for lack of a better term, but yet might be with regard to cancers, but might be associated with other age-related disorders. So we've been studying those mutations because we know, in fact, and this comes from publications that have been already out there in the literature, and you can't actually go to a conference now, whether it's a blood conference or a genetics conference, without seeing something about age-related clonal hematopoiesis and its potential impact on numerous diseases. But we know now that it explains a fair proportion of the variation for a number of disorders that are age-associated, not just cancers, but also things like type 2 diabetes, cardiovascular conditions, and sudden death. So we're largely interested in trying to understand why some of these people will develop a cancer, or why ARCH is a pre-cancer, why some people remain healthy, while some people develop no cancers but might develop another condition. So as I mentioned before, we have a number of programs in this space right now. We've been looking at single-cell RNA sequencing from participants in the Ontario Health Study, both from people that we consider to have healthy blood profiles, if you like, also from individuals that we think are ARCH-related. With SEGI, we're going to be doing deep targeted sequencing of blood profiles for a panel of genes that we know to be implicated in ARCH. And we actually now have a publication in review right now at Nature Communications with Kim Skeed on using this information to understand the evolutionary pressures associated with ARCH. So one of the projects that we have ongoing right now is as I mentioned before, where we're looking at individuals from the Ontario Health Study. This is Alyssa's project, where we've stratified the cohort into individuals that are old healthy, old unhealthy, young healthy, and young unhealthy based on CBC risk score that we've developed based on complete blood counts and grouped according to age as well, so that we can look at not just the mutations that are accruing in these different groups that may be associated with ARCH, also not necessarily associated with ARCH, and also look at their single-cell transcriptomic profiles. And so that project is well underway. We've completed over 400 individuals with regard to single-cell RNA sequencing. We can, parse those cells into the canonical cell types using canonical cell markers. And we can ask questions like, how do cell content change with respect to that stratification of individuals? So we know, for example, that older individuals, this isn't really new, tend to have more myeloid and NK cells. And that tends to be associated with age-related clonal hematopoiesis. ARCH contributes to a more myeloid bias in your blood pool. So we're capturing that, if you like, in our comparisons. So again, we have our risk groups here: young healthy, young unhealthy, old healthy, and old unhealthy here. And so we're seeing that myeloid bias in some of our age groups here. But what we're also capturing is, if we look at, say, our old our old healthy group here, for example, the orange bars here, is that older healthy individuals compared to their counterparts, say older unhealthy individuals, have more dendritic cells and for some reason have fewer B cells. So we're capturing interesting signatures in this in this way of stratifying this population cohort, stratified using this kind of approach to characterize the cohort with this, if you like, these 4 risk groups that we have here. Sorry, I'm raising my hands like this, but it's hard to do that with a Zoom call. Other things that we have ongoing with the ARCH process is not just looking at point mutations or gene expression patterns, but also looking at structural variants. Not all mutations are point mutations. They tend to also, much of the genome is littered with these structural variants, larger scale variants. And those structural variants also are associated with ARCH. And we're able to use the genotyping data that we have in the cohort, the Ontario Health Study, to capture structural variants that are occurring not in germline, but in somatic tissue, and how they might be associated with health outcomes like these clinical phenotypes. This is work that my postdoc Mary Jalouis has started and has presented a number of times at some of the international meetings. And so we're capturing patterns that are associated with different kinds of somatic structural variants, deletions, duplications that are occurring in blood, and they may be seeming to be associated with, say, cardiovascular diseases, like that's what a heart here means, and a C here is referring to things that are associated with cancers like WNT3. So we're just saying here that these somatic structural variants seem to be associated with gene expression of these types of genes. So again, we're capturing these age-related associations. Last thing I wanted to talk about in the ARCH space here is work that we're doing to evolutionary model the impact of somatic mutations in blood. So this is work done by Kim, as I was mentioning before, that we already have now in review. And we've been using empirical data where we've captured at from high coverage sequencing data mutations from individuals who were either not diagnosed, but they still have crude somatic mutations, versus individuals who were diagnosed with a cancer. That's what the red bar here means. So there's a somatic mutation occurring perhaps here in the early stages of the blood. And looking at those mutation profiles from the empirical data and comparing that to simulations or in silico data for different evolutionary processes. That's what's here on the right. And we've developed a number of tools, in Kim's case, we developed a neural network where we can use this information to train data sets so that we can look at summary statistics for those mutational profiles and then ask which evolutionary model does that data fit? So the data that we're looking at is data associated with a paper that we were part of, but was led by Sagi, John Dick, and Laurent Schluch. So Sagi's the first author in this paper. And in this paper, they were able to show that somatic mutations occur, don't know why that's happening, somatic mutations are occurring in both control individuals, people who didn't develop acute myeloid leukemia, but also accumulated somatic mutations in pre-accumulated acute myeloid leukemias, and in fact at higher frequencies. And everything you're looking at is that the variant allele frequencies are higher in blood, the numbers of somatic mutations are higher in blood for those pre-AMLs, and as well the mutations associated previously associated with either leukemias or other cancers are higher in blood in those pre-AML individuals. So we've been modeling that data. I'm going to move forward a little bit more quickly, such that we can then ask or test whether these various models here on my right fit the data, right? So does the mutational spectrum in those individuals that are either pre-AML or those individuals who don't develop AML, do those mutational spectrum fit these models of evolution, whether positive selection, like drivers driving a mutation or a clone is driving the growth of those clones, negative selection, combinations, so mutations are keeping cells at a lower frequency, combinations of the two, or having no impact at all, neutral, or just genetic drift, for those who remember that from population genetics. So, sorry, Segi, I skipped over your picture really quickly. So Segi's, again, really driving that data that we were looking at. So we have summary statistics that look at numbers of mutations, the annotations of the mutations, the allele frequencies of those mutations. Simulations were largely developed by Armand as part of her thesis project as well. And we've developed summary statistics, and we compare the summary statistics using a neural network. And I'm going a little bit quickly here. This is just to highlight an ensemble approach that we use in this neural network to be able to ask whether those summary statistics best fit these models of evolution. This just gives you a really quick picture of how well this does. This is probably not even the most recent picture, but it's actually improved since then. But even just from the simulations, we can see that in our confusion matrix here, we get a really nice comparison between the predicted class relative to the true class. So from simulations in silico, we know what the simulation was trying to tell us, and we can use the simulations to test against itself to give us a sense of how well our approach is working. And you can see it's nice and red along the diagonal, which means that we're getting what we should be getting in silico. And then we can apply that to data. And what we see with the somatic mutations, of course, is that, oh, we're already at 25 minutes past the hour, is that blood either largely fits either a combination class where both damaging and positive mutations fit the data, or in both cases, you'll see in both the controls and the pre-leukemics that both negative mutations and no necessary signature of positive mutations can be observed as well. We can break that down by age as well, which is what we're seeing here with this color scheme up here. And you can see that both in the controls and the pre-leukemics, you see that there's a consistent models where it's unlikely that you'll ever get a situation where the that positive mutations are arising solely. In fact, most of the time, a positive mutation is largely landing in a clone in the background of a negative mutation as well, this combination class. And we see that accruing with age. Okay, so for the purpose of time, I want to move on to what we're doing in the solid biopsy space, this project too, because I haven't really had a chance to talk about it, but some of this work is really exciting. So what are we doing here in this space? Well, here we're looking at cell-free cancer early detection methods. And here what we're trying to do is use plasma cell-free DNA to capture incident cancers in the context of solid tumors as well. And here what we're trying to do is we're trying to see if we can detect a solid tumor signature in plasma with cell-free DNA. And some of the questions that we have here, sorry, I don't know why my mouse is doing this, that we have are trying to understand not just whether that cancer is coming up, tissue of origin, and what are the molecular features of these early cancers. And we're really trying to move into this space where how early can we go in terms of our detection? I see you, Michelle, and I'll be just using the last few minutes here. So we're looking at cell-free DNA. And you know what happens with cell-free DNA when you have a tumor is that tumors, you have those tumors during that growth process, they will die, and when they die, they shed DNA, and you capture that in the cell-free space. And what we've done, what Nick has done, is he's developed both a mutation panel and an approach to looking at methylation, and I'm going to be mostly focusing on the methylation signatures in the cell-free DNA space here. And I'm just going to move forward to that space. Largely the cell-free methylation signature that we've been looking at is an approach that Daniel D. Carvalho has developed, where in plasma, we can detect, or he can detect, signatures that are specific at time of diagnosis to, you can see them here in this pretty poor resolution figure, pancreatic, lung cancers, breast cancers, and so on. So what we've been doing is we've been trying to use that approach in those early cancers that were captured before somebody was diagnosed with a cancer. Lots of QC goes into this space. What we do in those methylation approaches, we're capturing cell-free DNA from plasma, enriching for the methylated regions. And you can see here that this is all work being done by our genome technologies at OICR. So big thanks to them for helping with this protocol. These are the cancers that we've been looking at so far. We've been looking at these breast cancers. You can see that some of these breast cancers date back to 2010, and the diagnoses were as recent as 2016 to 2018. And we've been looking at pancreatic cancers as well. We have information on those breast cancers and pancreatic cancers with respect to stage as well. And so the work that Nick's been doing is really focused on capturing differential DMRs, which are differentially methylated regions in these pre-cancers compared to control. So for every cancer that we have an age and gender or sex matched control. This is the model he's been using to capture that information that you have here. I'm not going to have time to get into that here. You see these differentially methylated regions. These come up as volcano plots, as you like. And so, you know, this is just some early data that I wanted to show you here. And this is, Michelle, this is, I think, my second or third last set of slides, so that I'm going to have time, I think, to present here. What you can see here is that breast versus controls are really parsing really well. So 2 years before that person came into a clinic, we're able to capture with a methylation signature in the sequence data, a signature already of development of that cancer. And you can see that for 3 years, and you can see that for 4 years as well, right? Now, what's interesting as 4 years and later progresses, it starts to get a little noisy. And that actually might be biologic. That might actually have something to do with the quality of the data, but you start to see a little bit of overlap here because it may be that there isn't a signature in that participant as early as 4 years before that cancer. So that seems entirely reasonable with regard to how this is progressing. So some things that Nick's been also looking at here is looking at how the regions, the methylated regions in the genome are overlapping between, say, 2 years, 3 years, 4 years, 6 years, 5 years, and 6 years. Generally, there's pretty good overlap in terms of which regions are methylated. The fun part about this program, and sorry, Morgan, this really is getting to my last couple of slides, is that from the controls, you get a lot of other information. You got information about smoking history. You got information about weekly alcohol consumption, and it's manifesting itself in these methylation signatures here as well. So you can get a signature of that as well. So you're able to use these types of approaches to look at other comorbidities that might be in the population. And so you get these nice kind of comparisons here as well. And lastly, the last thing I wanted to mention is pancreatic cancers. So early days here still in terms of sample sizes, but once again here, you've got pancreatic cancers that are separate. I don't know what's going on here. Pancreatic cancers here that are separating nicely from our controls as well. We don't have as many pancreatic cancers as a rare condition in our cohort. We only have a total of 17 incident cancers in the Ontario Health Study. So, you know, this isn't going to grow too much more in size, but you can already see that we're doing pretty well in that space as well. So other things that are ongoing right now are Cox survival analyses to look at time to diagnosis, capture, say something more about the annotations for these methylated regions, see how well we can do in terms of, you know, if a participant comes in, can we really say that the tissue, like we know where these cancers are, so the analyses are easier, but when a patient comes into a cohort or into a clinic, you don't necessarily know where the cancer is. So we want to be able to use these tools to be able to identify the tissue of origin. And there's my last slide. So a whirlwind tour of some of the stuff that we're doing in the AO in the first project. So thanks for your